Good morning, everyone. Today we are going to discuss one of the important and the highly relevant topics in the pharmaceutics and the novel drug delivery systems, that is, controlled drug delivery systems, commonly abbreviated as CDDS. So, controlled drug delivery is not simply about making a dosage form release a drug slowly. So the fundamental objective is much broader here. It is about controlling how much drug is released, when it is released, and sometimes where it is released. So conventional dosage form generally produces fluctuations in the drug concentration. So after administration, what will happen? So the concentration rises gradually here and reaches a peak and then gradually decreases. So when the concentration falls below the therapeutic level and the dose is required here. So but whereas in case of controlled delivery, so controlled delivery attempts to overcome this problem by designing a formulation that provides a predictable and a controlled drug input over a predetermined period. So as a result, what will happen? The drug is continuously supplying to the bloodstreams here. Okay, so the drug level is maintaining constant here. Okay, so that is the main aim of this controlled delivery systems. So here we are visualizing the doses from containing the drug. Instead of releasing the entire dose immediately, so the formulation is engineered so that the drug molecules are released according to the particular mechanisms. So they may adopt different mechanisms. So the mechanisms may involve diffusion, dissolution, erosion, swelling, osmotic pressure, ion exchange, degradation of polymers, or response to the external or physiological stimulus. Okay. So at the M-Pharm level, we should not look at CDDS merely as a formulation technology. So it is an integration of physical pharmacy, polymer science, pharmaceutics, pharmacokinetics, mathematical modeling, formulation technology, and increasingly nanotechnology and personalized medicines. So throughout this lecture, we will move from the basic concept of the control delivery to the advanced mechanisms, mathematical models, evaluations, applications, limitations, and the future perspectives. So concept and the definition. So let us first understand exactly what we mean by a controlled drug delivery systems. So a controlled drug delivery system is a formulation or a device designed to deliver a drug at a predetermined and a controlled rate for specific period of time, with the objective of maintaining the drug concentration within the desired therapeutic range here. So look at the graph. The conventional system produces the significant fluctuations here. Following administration, the plasma concentration increases rapidly. It may reach a high peak and then decline. So repeated administration produces repeated peaks and thoughts. So in contrast, a controlled delivery systems attempts to produce more predictable drug concentration profile here. So an important examination point is the difference between the sustained release, controlled release, and the extended release. So here is a little bit confusion with these three terms here. So let me break the confusion here. So extended release formulations generally prolong the drug available compared with the immediate release dosage forms. Okay, but whereas in case of sustained release refers broadly to maintain the drug release for a longer duration here. Okay, and whereas in case of controlled release has more specific implication. So the release rate is designed to be controlled and predicted. So we should not automatically use the terms sustained release and controlled release as a perfect synonyms here. Okay, so the ideal system should provide predictable drug release, producible performance, adequate therapeutic duration, minimum concentration fluctuations, reduced dosing frequency, and improved patient compliance and acceptable safety. Okay, so now the important question is, why do we actually need the controlled drug delivery? Okay, so let us compare the conventional therapy with the controlled delivery here. So in conventional dosage forms, particularly immediate release dosage forms, the drug concentration can fluctuate significantly here. So we may experience a peak concentration soon after administration, followed by a decline. So if the peak became too high, adverse effects may occur. So if the concentration becomes too low, therapeutic efficacy may be lost. So we have a fundamental problem. So peak related toxicity and throw related therapeutic failure. So controlled delivery attempts to minimize these fluctuations, and the major objectives are maintaining the therapeutic concentrations, reducing the dosing frequency, improving the patient compliance, and reducing the adverse effects associated with the high plasma concentration here, and providing a prolonged pharmacological effects. Okay, so imagine a patient who needs a medicine even for every 4 hours. So that means the patient must remember 6 doses in a 24 hours. So if we can formulate the same drug to release over 12 to 24 hours, the dosing burden can be substantially reduced here. Okay, so that's the importance of this controlled release. So however, the controlled delivery is not automatically superior for every drug. Okay, so a drug must possess a suitable properties such as appropriate half-life, therapeutic index, dose, adsorption characteristics, and the stability. Okay, so to understand why maintaining the concentration is important, we need to understand the therapeutic window and pharmacokinetic principles here. Okay, so therapeutic window and the pharmacokinetic basics. So this is one of the most important conceptual slide here. So therapeutic window is the concentration range between the minimum effective concentration or MEC and the minimum toxic concentration or MTC. Okay, so below MEC the drug may not produce the sufficient therapeutic activity here. So observe clearly the graph here. So above MTC the probability of the toxicity increases. So the ideal objective is to maintain the drug concentration within this therapeutic window. Okay. Here we can see concentration on the vertical axis and the time on the horizontal axis. So an immediate release product may show the sharp peaks and the valleys here. So a control system attempts to flatten these fluctuations. So observe the graph. So now let us connect this with the pharmacokinetics. So the performance of a controlled delivery system depends mainly on ADME. That means absorption, distribution, metabolism, elimination, and half-life, clearance, and the volume of distribution. Half-life. For example, if a drug has an extremely short half-life, maintaining the therapeutic concentration becomes more challenging here and may require a high release rate or a alternative delivery strategy is required here. On the other hand, if a drug already has a very long half-life, controlled release may provide a limited additional benefit here. Clearance. So clearance tells us how efficiently the body eliminates the drug. So if clearance is high, the system needs to provide the adequate drug input to compensate for elimination here. So volume of distribution. So volume of distribution influences how extensively the drug distributes into the tissues and therefore affects the plasma concentrations. Therapeutic index. So another important consideration is the therapeutic index. So a drug with a narrow therapeutic index requires particularly careful control of drug exposure. So controlled delivery designs should never be separate from the pharmacokinetics. So the formulation scientist must design the release profile according to the pharmacokinetic requirements of the drug here. So classification of the controlled drug delivery systems. So now we move to the classification. Controlled drug delivery systems can be classified according to the mechanism responsible for the drug release here. And first one is diffusion-controlled systems. So the drug molecules diffuses through a polymeric membrane or a matrix here. So these can be further divided into reservoir systems and their matrix systems. And second one is osmotically controlled systems. So the drug release is controlled by osmotic pressure here. So water enters the system through an semi-permeable membrane generating the pressure that drives the drug solution or suspension through an outlet. Okay, so that's the mechanism here. And next one is ion exchange system. So the drug molecules interacts with an ion exchange regime and subsequently exchanged with the polymeric ions here. Okay, and next method is the erosion control systems. So the polymers the matrix gradually erodes, allowing the incorporated drug to become available here. And next method is the stimuli-responsive systems. So these systems respond to a specific stimulus. The stimulus may be pH, temperature, enzymes, glucose, magnetic field, light, ultrasounds, or electrical stimulations. Swelling control systems. The polymer absorbs the water and swells. So this changes the diffusion pathway and controls the drug release. Okay, that is about the swelling control systems. So in real formulations, mechanisms are often not completely independent. For example, a matrix tablet may involve the water penetration, polymer swelling, diffusion, erosion simultaneously here. Okay, principles and the drug release mechanisms. So before discussing individual systems, so let us understand the fundamental principles here. So the drug release occurs when the formulation creates a pathway through which the drug can move from the dosage form into the surrounding biological environment here. So major mechanism we can broadly consider diffusion, dissolution, erosion, swelling, osmosis, ion exchange, degradation, like that. So diffusion means movement of a drug molecules from higher concentration to the lower concentration. Dissolution means the drug dissolves from a solid matrix or coating here. Erosion means the carrier material is gradually removed. So that's about the erosion technique. And swelling, polymer absorbs water and increasing in the volume, and modifying the drug diffusion is observed here. And osmosis, so osmotic pressure drives the drug release, and ion exchange, the drug is released through exchange with the ions in the surrounding media. In degradation, the biological or biodegradable polymers break down and release the incorporated drug. So the formulation scientist selects the mechanism according to the drug and the therapeutic objectives here. For example, if we want the highly predictable delivery, an osmotic system may be used. If biodegradability is important, a biodegradable polymer systems may be preferred here. So if a physiological feedback is required, a stimuli-responsive systems may be investigated. So like that we have to choose the method. Diffusion control systems. So the diffusion control systems are among the most extensively studied control release systems. So there are two major approaches here: reservoir systems and the matrix systems. We already discussed. So reservoir systems. In a reservoir system, the drug is located in a central reservoir. So the reservoir is surrounded by a polymeric membrane, and the membrane acts as a rate controlling barrier here, and the drug must dissolve and then diffuses through this membrane. So that's the simple mechanism. So imagine the drug in the center, and the surrounding membrane controls how quickly the molecules can leave. So if the membrane permeability and the thickness are properly controlled, we can obtain a predictable release profile here. So the release mainly depends on membrane thickness, diffusion coefficient, drug solubility, partition coefficient, surface area, and the concentration gradients here. Matrix system. So here the drug is dispersed throughout a polymeric matrix. Okay. So in reservoir system, what we did, the drug is placed in the center. But whereas in case of matrix system, here the drug is mixed along with the polymeric matrix here. So when the formulation contacts the release media, the fluid enters the matrix here, and the drug dissolves and then diffuses through the polymeric network. Okay. So that's the simple mechanism here. Difference. So the major difference is reservoir. The drug core plus rate controlling membrane is seen, and whereas in case of matrix, the drug distributed throughout the polymeric material here. So that's the main difference. So reservoir systems can potentially achieve high concentration release. Okay. So limitations means, however, if the membrane develops a defect or ruptures, dose dumping can become a corner here. Okay, so that's the major limitation of this system. Mathematical basis of the diffusion. So now we come to an important inform level concept, the mathematical description of the diffusion. Fick's first law. So the fundamental relationship is represented by Fick's first law here. So observe the equation, J is equal to minus D into dc by dx. So the equation tells us that diffusion flux is proportional to the concentration gradient here. So here J is the diffusion flux, D is the diffusion coefficient, C is the concentration, and dc by dx represents the concentration gradient here. So the negative sign indicates that the diffusion occurs from higher concentration towards the lower concentration. So if the concentration difference is large, the driving force for the diffusion is greater here. Suppose the drug concentration inside a formulation is high and the concentration outside is low. So there is therefore a concentration gradient here. So the drug molecules move down this gradient. So factors affecting diffusion. So the diffusion coefficient depends on molecule size, polymer structure, temperature, viscosity, and the interaction between the drug and the polymer here. So for membrane control systems, we also consider the membrane thickness and the permeability here. So a thicker membrane generally increases the distance that the drug must travel. So therefore the membrane thickness can be an important formulation variable here. Okay, so matrix systems, advanced concepts here. So matrix systems are particularly important because they are widely applicable to the oral controlled release dosage forms here. So hydrophilic matrix, hydrophilic polymers such as HPMC can form in a gel layer when exposed to the aqueous media here. The sequence is water penetration, polymer hydration, gel formation, drug dissolution, and erosion, or maybe erosion. Okay, so initially the water penetrates through the outer region and the polymer hydrates and form a viscous gel, and the gel creates a diffusion barrier. As time progresses, the gel layer becomes thicker and the drug diffuses through this hydrated polymer here. Eventually, the polymer erosion may contribute to release. Next is the hydrophobic matrix. So in hydrophobic systems, the materials such as ethyl cellulose or waxes creates a water resistant barrier here. So the drug release occurs primarily through the pores, channels, or the diffusion pathways. So critical formulation variabilities includes, so important variabilities are there, that is polymer concentration, polymer viscosity, drug solubility, particle size, compression force, porosity, and the polymer drug interaction. So all these factors are needed to be considered. So therefore a matrix formulation is not simply a matter of selecting a polymer here. We must understand the relationship between the polymer properties, hydration behavior, microstructure, and the drug transport here. So now we move to one of the most elegant controlled release system, that is osmotic drug delivery system. So the fundamental principle is osmosis here. We all know that. So the water moves into the system through a semi-permeable membrane, and the system contains an osmotic agent. As water enters, the internal pressure develops here. So this pressure pushes the drug formulation towards the delivery orifice. It is at bottom. So the basic components include semi-permeable membrane, osmotic agent, drug compartments, a delivery orifice. So notice the semi-permeable membrane here. So it allows the water to enter but resists the movement of the drug and the osmotic solutes here. So once the water enters, the internal osmotic pressure increases obviously. So this drives the drug containing formulation towards the outlet. So advantages means, one of the most major advantages is that the drug release can be relatively independent of the several physiological factors. For a well-designed system, release can be more predictable than the conventional diffusion systems here. Formulation variabilities includes membrane permeability, membrane thickness, osmotic pressure, orifice dimensions, drug solubility, and osmogen concentrations. So commercial osmotic pump technologies have demonstrated how osmotic principles can be converted into the sophisticated oral drug delivery systems here. So the important point is that osmotic systems use water and osmotic pressure as the driving force for controlled drug release here. Okay. Dissolution, erosion, and swelling systems. So this slide compares three mechanisms. Dissolution control. So in dissolution control systems, the drug release depends on dissolution of the drug or surrounding coating or matrix. So if the dissolution rate is slow, the release is prolonged here. Okay, that's about the dissolution control. In case of erosion control, in erosion control systems, the polymers or the matrix gradually disappears from the surface or bulk. So as the matrix erodes, the drug becomes available here. Okay. Example of mechanisms. Erosion can be a surface erosion or a bulk erosion. Surface erosion occurs predominantly from the outer surface, and bulk erosion involves degradation throughout the polymer matrix here. Swelling control. So swelling control systems use polymers capable of absorbing the water here. So water enters the polymer, causing it to swell. So this produces a hydrated network through which the drug molecules diffuse. Okay, so that's a mass definition. So comparison. So remember, dissolution means a material dissolves, erosion means a carrier is removed or degrades, swelling, polymer take up water and changes the structure. Okay, this is how we will remember the concept. So in many practical formulations, these mechanisms overlap here. Okay, so by heart all these mechanisms. And the next one is ion exchange resin systems. So in ion exchange systems, we use a reversible ionic interactions between a drug and an insoluble resin here. So that's the basic principle. Means suppose the drug is positively charged, so it can interact with the negatively charged functional group present on the resin. Okay, so the drug becomes bonded to the resin when the drug resin complex enters physicochemical system, that is ions such as sodium, potassium, or hydrogen ions can exchange with the drug here. Okay, so this release the drug finally. So initially the drug molecules are associated with the resin, and then physiological ions compete for the ion exchange sites, and the drug is gradually displaced. Okay, factors affecting release. So the release mainly depends on several factors like ionic strength, pH, resin particle size, degree of the cross linking, drug resin affinity, and composition of the surrounding medium here. Okay, applications includes these systems can be particularly useful when the prolonged release and the taste maskings are desired. Okay, so therefore the release mechanism here is not a simple diffusion, so it based primarily on the reversible ion exchange. Okay, and next method is the biodegradable control delivery. So biodegradable control delivery is particularly important for the modern drug delivery here. So here what's the main principle means, the drug is incorporated into a biodegradable polymer here. So the polymer gradually undergoes degradation. As the polymer structure changes, the drug is released. Okay, that's the simple mechanism involved in this process. So common polymers includes PLA, PLGA, PCl, and other biodegradable polymeric systems are included. So PLGA is particularly important in pharmaceutical drug deliveries. So it is composed of lactic acid and the glycolic acid units. So the ratio of these components can influence the degradation behavior. Release mechanisms. So the drug release from the biodegradable systems may involve diffusion, polymeric degradation, polymer erosion, or a combination of these. Okay, so these systems are mainly used in the microspheres, nanoparticles, injectable depots, implants, and long-acting drug delivery systems. So polymeric degradation kinetics therefore become an important factor in the formulation design here. So we must understand both drug diffusion and the polymer degradation here. Okay, stimuli-responsive drug delivery. So now we come to more advanced area here, that is stimuli-responsive drug delivery. So these systems are sometimes described as the intelligent or smart drug delivery systems. We already discussed some systems in previous videos. So because they respond to the specific stimuli. So internal stimuli includes pH, temperature, enzymes, glucose or redox conditions. Example, pH. So a formulation may be designed to remain suitable in one pH environment but release the drug in another. So this principle is particularly relevant to the gastrointestinal targeting here. So glucose responsive systems. So glucose responsive systems have attracted interest in diabetes related drug delivery. So the system attempts to respond to changes in the glucose concentrations. External stimuli includes magnetic field, light, ultrasound, electrical stimulations. So general mechanism. So the sequence can be summarized here, that is stimulus, carrier responses, structural changes and the drug release. So that's the simple sequence here. So this concept moves the drug delivery from passive release towards the responsive and the potential personalized therapy here. Okay. So factors affecting drug release. So this is important formulation development slide. The drug release is influenced by three broad categories here. So the drug related factors includes dose, aqueous solubility, molecular weight, particle size, diffusion coefficient, partition coefficient and stability. So first factor is solubility. So drug solubility is particularly important. A higher soluble drug may dissolve rapidly, while a poor soluble drug may become dissolution limited. So polymer related factors includes molecular weight, concentration, hydrophilicity, porosity, cross-linking, swelling and degradation rates. Physiological factors includes gastrointestinal pH, gastric emptying, gastrointestinal motility, food, fluid, volume, enzymes and the disease conditions. So when we observe a particular release profile, we should not immediately blame the polymer here. So when we observe a particular release profile, we should not immediately blame the polymer here. The final release profile is the result of the interaction between the drug properties, formulation properties and the physiological conditions here. So now we enter one of the most important analytical area in the controlled drug delivery, that is release kinetics. Mathematical models help us understand the mechanism of the drug release here, that is zero order kinetics. In zero order release, the amount of the drug release per unit time remains approximately constant here. So the mathematical representation is, the mathematical relationship is Q is equal to Q0 plus K0 T. So the important feature is that the release rate is constant here. So this is often considered an ideal target for the controlled drug delivery here. So next one is the first order kinetics. In first order release, the release rate depends on the amount of the drug remaining in the dosage form. So as the amount remaining decreases, the release rate obviously decreases. Higuchi model. The Higuchi model is commonly associated with the diffusion-controlled release from matrices. So the basic relationship relates the amount released to the square root of time here. So a plot of cumulative amount released versus square root of time can help assess whether the system follows the Higuchi type behavior or not. Okay. And next one is Course, Mayer and Peppas model. So this model is useful when the mechanism is not clearly represented by a single classical model. So the release exponents provides the information about the mechanism here. So model fitting alone does not prove a mechanism here. So a high correlation coefficient does not automatically mean that the proposed mechanism is scientifically correct. So we should consider the formulation structure, polymer behavior, experimental conditions and the multiple models here. Okay. Course, Mayer, Peppas model. So now let us explain the coarse Mayer and the Peppers model in more detail. So the equation is commonly expressed as mt by m infinity is equal to k into t power n. So here mt is the amount released at the time t, and m infinity is the total amount released, and k is the kinetic constant, and t is time, and n is the release exponent here. So this is about the equation. So for meaning of n, for a cylindrical system, the value of n provides the information about the release mechanism here. So when the n is around the Fickian region, the diffusion is the dominant mechanism here. As an n increases in the intermediate region, so we may observe the anomalous or the non-Fickian transport here. So this indicates that more than one process may contribute here, such as diffusion combines with the polymeric relaxation or swelling. So at still higher n values, case 2 transport and the related mechanisms may become important here. So the exact interpretation of the n depends on the geometry of the dosage form. So we must never memorize our one universal range and apply it to every system. Okay, so graph, the slope of the logarithmic plot gives the release exponent here. So this makes the model particularly useful for the mechanistic interpretations during the formulation development. Okay, in vitro evolution and the IVIVC. So after developing a controlled release formulation, we need to evaluate it. So in vitro evaluation includes drug content, content uniformity, physical properties, in vitro drug release, release kinetics, and stability. Okay, first one is the dissolution testing. So the dissolution testing is particularly important here. So we collect the samples at a predetermined time intervals, we analyze the amount of the drug released, so then we plot a cumulative percentage drug release against the time. So the profile tells us whether the formulation produces the immediate, sustained, delayed or controlled release. IVIVC. So now we come into in vitro in vivo correlations or IVIVC. So the basic objective is to establish a relationship between the drug release measures in the laboratory and the drug absorption or performance in the body. So why important? Why it is so important means, if a meaningful IVIVC is established, in vitro dissolution may provide useful information about the expected in vivo behavior here. So this can support the formulation development and potentially reduces the need for the extensive repeated in vivo studies during certain development stages. Important destinations. So however, IVIVC is not always achievable. So the relationship may be influenced by absorption, metabolism, gastrointestinal transit, and the formulation differences and the physiological variabilities. Okay, is it clear for everyone? So next, move on to the applications and the delivery platforms. So controlled drug delivery technologies are not limited to one dosage form here, I already told. So oral systems, these includes controlled release tablets, matrix tablets, osmotic systems, and the coated systems. Transdermal patches can provide a prolonged drug delivery across the skin. So microspheres and nanoparticles. Microspheres and the nanoparticles provides here opportunities for the controlled and the targeted delivery here. Liposomes are vesicular carriers capable of incorporating the hydrophilic and the lipophilic substances depending on the structure. Injectable depots. Long-acting injectable depots can provide the prolonged drug exposure and the release during the frequency. Long-acting injectable depots can provide the prolonged drug exposure and reduce the dosing frequency here. Implants can provide the drug release over weeks, months, or longer depending on the design. Ocular systems. So ocular control delivery attempts to overcome the rapid clearance of the conventional eye depots here. Emerging future platforms includes smart polymers, nanotechnology, personalized medicine, stimuli-responsive systems, and AI-assisted formulation designs. Advantages, limitations, and the future perspectives. So let us summarize the major advantages here. So controlled drug delivery can provide the reduced dosing frequency, improve the patient compliance, and prolong the therapeutic action. So reduce the plasma concentration fluctuations and improve the therapeutic efficacy here, and potentially reduces the adverse effects. So all these are all the advantages. So but we should also recognize the limitations also. Controlled release formulations may involve higher manufacturing cost, greater formulation complexity, difficult dose adjustments, and risk of dose dumping, dependence on the drug physicochemical properties, and the challenges in the scale up. So all these are other limitations. Future perspectives. So the future of the controlled drug delivery is moving towards the precision and the personalization. So smart polymers can respond to the specific stimuli. Nanotechnology can improve the targeting and the delivery here. The personalized medicines can potentially match the drug delivery characteristics to include individual the patient needs here. So artificial intelligence and the computational modeling may help predict the formulation behavior and optimize the formulation variabilities here. So the stimuli objective is not simply to slow down the drug release. So the real objective is to control the drug exposure in a scientifically predictable manner and achieve the desired therapeutic outcomes. Okay. So let us conclude today's discussion. So the controlled drug delivery systems represents an important area of the modern pharmaceutics because they allow us to move from the conventional repeated dosing towards more predictable and prolonged drug exposure here. So we discussed the several major mechanisms includes diffusion, dissolution, erosion, swelling, osmosis, and biodegradation and stimuli-responsive release. We also discussed the importance of the mathematical modelings through zero order, first order, Higuchi, Cosmeyer, Pepper's models. So, however, the mathematical fittings should always be supported by a proper understanding of the physical models or mechanisms. So, we also look at the formulation factors like physiological factors, in vitro evaluations, and IV, in-vivo, in-vitro correlations. So in simple words, the goal of the controlled drug delivery is to deliver the right amount of the drug at the right rate for the right duration and whenever possible at the right site. Okay. And as a pharmaceutical research moves towards the personalized medicines, nanotechnology, smart polymers, and the responsive systems, controlled drug delivery will continue to play a major role in the future of the therapeutics. So thank you for everyone for your attention. I hope this lecture has helped you understand not only what controlled drug delivery systems and but also why their design requires an integration of the pharmaceutics, pharmacokinetics, polymer science, and the mathematical modelings. Thank you.